Hey there, AP Bio. Here is the second set of notes for Chapter 11. Remember that the first set of notes had to do with cellular communication, and we said that in a signal, that there's a reception, that there's a transduction pathway, and there's a response. And so the to the last set of notes we talked about the reception. In this set of notes, we are talking about the transduction pathway. Our third set of notes, we're going to be talking about the response. So let's get to this. So cascades of molecular interaction are multi-step pathways, which means that they involve many molecules. And so the benefits of this, one is that the message can be amplified, meaning that it can grow in the number of molecules that are being used pretty quickly. So if we look at this picture here, if this is epinephrine, which is a signal, it's going to activate a receptor protein, which then is going to activate, let's just say, three adenylyl cyclases. And each of those adenylyl cyclases are going to be able to activate multiple cyclic AMPs, and so on and so on and so on. So this number grows and grows and grows. And this allows for a response to occur quickly and effectively and efficiently. It also allows for, so the fact that it's a multi-step pathway, or that there are multi-step pathways in responses, in signals, is that it allows for coordination. And we will talk more about that in these notes and in the next set of notes. But so multiple responses can occur at once. So here, let's talk about the transduction pathway. The message is passed on to a series of relay proteins. So here's our signaling molecule, here's our receptor protein, which we talked about last class. And here are our relay molecules, or our relay proteins. So oftentimes these are kinases, and remember that kinases are proteins that phosphorylate, and they phosphorylate other proteins. And the amino acids that can receive a phosphate is are the tyrosines, the serines, and the threonines right here. And so a protein has to have one of these available to receive the phosphate. And so if we follow this along, we can see that the relay molecule has been activated. And what it's going to do is it's going to activate this protein kinase. And so this protein kinase is going to take the phosphate from this ATP, so it turns it into an ADP, and it's going to activate this second protein by giving it a phosphate, either on the tyrosine, the serine, or the threonine. And we can see that that's going to happen again. An ATP becomes an ADP. But in this case, the phosphate is added to this protein here. And so 2%, about give or take 2% of our genetic code is thought to be instructions on how to build kinases. So they are very abundant in our cells. They have lots of jobs, and they are very important. So our next slide, we're going to look at this section right here in the transduction pathway. So equally as important as the kinases are the protein phosphatases. These deactivate, or they remove the phosphate. So here you can see, here's our phosphatase. So here's our active protein kinase, which is creating a signal or creating a cellular response. And so it's going to remove this phosphate, and it's going to inactivate this. And so this is really important because you need to be able to turn off a signal just as quickly as you turn it on. And so this removal of a phosphate is called dephosphorylation. And so transduction, not all components of a signal transduction pathway are proteins. We also have second messengers. So second messengers are the same thing as relay proteins, except that they are small ions or non-protein molecules that diffuse through the cell because they are water soluble. So if we call these relay proteins that are not, or these relay molecules that are not proteins, we call those second messengers. then we would call the signaling molecule like a first messenger, but we'll just call those signaling molecules. 3 examples of second messengers that we're going to use are cyclic AMP or CAMP, and we are going to investigate the effects of this in cholera, the disease cholera. We're going to look at cyclic GMP or cyclic guanosine monophosphate. Cyclic AMP is adenosine monophosphate, so 1 phosphate. So that's 2 less than the triphosphate. So again, the cyclic guanosine monophosphate, and we're going to look at the effects of that in Viagra, and then we're going to finally look at calcium ions. They're abundant on the outside of cells and in organelles such as the ER, the endoplasmic reticulum, the mitochondria, and the chloroplast. They're not very abundant in the cytosol. So if we look here, we can see that our relay proteins are doing the same thing, or these second messengers are doing the same thing as these relay proteins. So the ATP is going to be converted into cyclic AMP. The adenylyl cyclase is going to chop off 2 of those phosphates and turn it into cyclic AMP, which can then elicit a response in the cell. So Earl Sutherland, we talked about him in the last class. He was the guy that discovered that glycogen that epinephrine causes the breakdown of glycogen without epinephrine ever having to enter the cell. He said that there's got to be something else going on, this transduction pathway. So he was looking for second messengers. He found that the binding of epinephrine, which remember is adrenaline, to a liver cell causes an increase in cyclic AMP in the cell. And initially, the cell receives a signal by the G protein coupler receptor, and this can amplify the message about like 20-fold in just a matter of seconds, which is another reason why life is just amazing. And as we talked earlier, the adenylyl cyclase is going to clip the 2 phosphates off of the ATP to make cyclic AMP. And so this signal doesn't last very long because there is an enzyme called a phosphodiesterase, which is going to convert the cyclic AMP into just AMP, which is adenosine monophosphate. And the active form is the cyclic AMP. So this is going to elicit a response, or it's going to activate another protein to elicit a response. And so when it's converted into AMP, it's no longer active. That's the deactivated form. And so there's a video here which breaks down this sort of pathway, and we can look at the ATP molecule and see adenylyl cyclase is going to chop off these 2 phosphates to make cyclic AMP, and we call it cyclic because it has this ring at the end over here. And then phosphodiesterase is going to add water and make it AMP. Sorry that my picture here is cutting that off. So we said that we're going to talk about cholera, and cholera has to do with cyclic AMP. Cholera is a bacteria that causes diarrhea. It can lead to severe dehydration because you're losing tons of water, like liters and liters of water. And if you're in a part of the world where you can't get enough water, it can lead to death. And this is often spread by unsanitary conditions or improper disposal of wastewater. And so the bacteria produces a biofilm, which we talked about in our last lecture, on the lining of the intestines. And this produces then a toxin. So one bacteria communicates to all of the other bacteria that we want to produce this toxin. And so it spreads the biofilm, and so then all of these bacteria replicate and produce the toxin. And this toxin, what it's doing is it modifies the G protein that is used to regulate salt and water secretion. So how much water is going into and out of the cell. So here, if we're looking at this cycle here again, what's happening is that the G protein cannot hydrolyze the GTP into a GDP. So remember that the GTP becomes a GDP and it deactivates the enzyme, in this case the adenylyl cyclase. So because it cannot be in the deactivated form, it doesn't turn off. This pathway continues going and it continues to elicit this cellular response. This cellular response is to keep releasing salt into the intestines. And so if you release salt, we understand osmosis, water will follow. And so therefore, the body is getting rid of, because this is stuck in this activated position, GTP can't be modified to GDP, or it can't be replaced by the GDP, then you're going to continuously lose salt and water. And so this is one way that a disease can be caused by an interruption in this pathway. Let's look at how using these pathways can help solve problems. Viagra is a medicine that uses second messenger cyclic GMP, or guanosine monophosphate. And so here's our pathway right here. When one cell, surrounding cell, releases nitric oxide, this cell here is going to receive it, and it's going to create a pathway using cGMP as a second messenger, which then is going to create a number of different responses, but one of those is to relax the smooth muscle. And to relax the smooth muscle, it also relaxes the muscles. That's the muscle that's found in the lining of the arterial walls. And so if the arteries are relaxed, or the muscles in the arteries are relaxed, it's going to dilate, and they're going to allow for more blood to travel through. If there's more blood traveling through the arteries, there's going to be more blood traveling through your entire body. And what this does is it helps with erectile dysfunction. Erectile dysfunction is when the penis will stay flaccid even when you don't want it to. And so if you take Viagra, it's going to put more blood into all of the spaces in the body, including the penis, and so therefore you can have sex. And so what happens, what is this cyclic GMP, what Viagra does is it allows this to stay on. It inhibits the hydrolysis of the cyclic GMP to, we have in our book it says GMP, but this is, which is cyclic, which is guanosine monophosphate. In this image here says triphosphate. So it jumped a couple of steps. I don't want that to confuse you. It's the same thing. But so essentially what happens is this stays on. It doesn't get turned off, which means that the smooth muscle continues to relax. And that means that the blood vessels will stay dilated until the medicine wears off. And so that is one way that we use it to help. Finally, let's look at calcium ions. Calcium is abundant in our extracellular fluid and in our blood, but it's not abundant in our cytosol. And it is actually more abundant than cyclic AMP. And it's used for cellular responses like muscle contractions, exocytosis, and cell division. It's more, we find it, like we said, in the extracellular space, in the fluid, but also in the endoplasmic reticulum. So the cells are going to, when it comes into the cell, it's going to oftentimes pump it right into the ER, into the mitochondria and chloroplasts. And so there's not a lot of calcium inside the cell, which we should remember from talking about nerves and impulses. And so when you have a small increase inside the cell, you're going to have a large percentage change. So a small increase in calcium is going to allow for a great change. And so when calcium is signaled to enter the cytosol, it's usually released from the endoplasmic reticulum. And there are going to be two other second messengers involved, IP3 and DAG.